from preparation to imaging, sources of artifact in transmission electron microscope and scanning electron microscope by Dr. Shaimaa Kashef, Assistant Professor of Histology, Faculty of Medicine, Tanta University, Egypt. Hello everyone, dear guests and colleagues. I'm glad that I got the great opportunity to participate in a great scientific conference, sharing and exchanging valuable scientific data. My research focus on many fields, but I am very interested in electron microscope, so I'm honored to be a part of this conference. Begin with this question. Have you got an electron micrograph with these defects? What are they called? Yes, they are artifacts. Artifacts make the researcher always confused about their origin and the interpretation of the image. So our agenda will be definition of artifacts, causes of artifacts, how to avoid them, and the importance of their identification. So what artifact? It's a damage caused by preparation techniques and can be easily confused with samples microstructure. Causes of artifact by the researcher or during processing and imaging. In the next slide, this compartment will appear the wrong technique done by researcher and while the other one, the how to avoid it. The reference of electron micrographs in this talk is represented by QR code that you can easily scan. Obtaining the sample, some researchers take large samples that hinder the penetration of the fixed tip. So, your sample size should be 1 millimeter cubic. The second mistake is little fixed tip in relation to the total volume of the tissue, and the solution is excessive volume of fixed tip in relation to the total volume of the tissue is mandatory, which is 1 to 20. If you put excessive tissue in the tube with glyceraldehyde, your fixative can't reach all the tissue compartment. So the tissue on the left half of the image remain unosmogated as the osmium didn't penetrate deeper to the tissue. Improper handling of the friable organs such as the brain may cause artifacts, so perfusion is the best for friable organs. Also, some researchers left the sample in the air, so dryness of the sample is okay. You should rapidly transfer the sample to the fixed. Preparation artifacts. Fixation artifacts. Fixation artifacts. Immersion in saline before fixation can produce osmotic changes within the tissue that can cause edematous swelling of the cell. So the best maneuver is fixation is the first step before saline washing to avoid disruption of the tissue natural osmotic balance. In this electron micrograph, we can see the edematous swelling of the endothelial cells. Also, the use of inappropriate preservative instead of glyceraldehyde can also produce ultrastructural changes within the tissue. So the solution best for the transmission electron microscope fixation is 2.500% glyceraldehyde plus 2 to 400% PFA for 30 minutes to overnight. Here we can see disturbance of the heterochromatin of the nucleus and has become difficult to distinguish from the surrounding matrix. Myelin sheath is very susceptible to be disturbed during the transmission electron microscope fixation and need optimum fixation and careful tissue handling. Here we can see in this electron micrograph a splitting and evagination of the myelin sheath with axonal shrinkage, with optimum and ideal fixation process, where an ideal micrograph of the myelin sheath without splitting or evagination. Mitochondria are also affected by poor fixation. Poor penetration of the fixative cause empty of a portion of them. Moreover, they appear swollen with crystallisis, which may lead to misinterpretation of your data, not because the drug or disease that you're working on it, but due to poor fixation. Dehydration artifacts. Overhydration causes shrinkage of the tissue, which may give you a false impression and interpretation that red asterisk is a dilatation of the intercellular space, but actually it is caused by shrinkage of the cells due to overhydration. Here we can notice holes in the tissue due to failure to remove water before embedding. Impregnation artifacts. Here, holes in the tissue and the resin due to poor impregnation. Here we can see cracks in the resin due to improper polymerization of the resin. Sectioning artifacts. Sectioning process is an interactive process including four elements: knife and the block, angle of the section, and the speed of the cutting process. Alternation or disturbance to one element of them lead to bad quality of the section with holes and knife mark. So, how are artifacts produced during sectioning? It's common that dirt or oil on the face of a block being sectioned may diffuse out into the surface of the water in the pot and contaminate the section, the knife, and leading to knife contamination and pot contamination. Unfortunately, the water used for floating ultrasonic section can be often ignored and become a potential source of artifact. So, ultra pure water or boiled distilled water must be used to avoid contamination. Bacterial clusters are lying over the section due to contaminated water in the pot. When sections are cleaved from the block, they bend sharply over the knife and then upward to the surface of the water. Cracks and tissue damage may occur. Also, the edge of the knife may be too dull or the cutting process may be very rough. All previous causes leads to sectioning artifact. As I said before, that knife contamination may be result from dust that bind to the knife edge and become a permanent part of the edge of the knife. So the solution will be continuous polishing to remove this particle and routine cleaning with wipes along the edge after every day of use and wash every week. Only diamond knives are at risk since they may be retained on surface for several reasons. Here in this electron micrograph, knife mark in rat's liver section can be noticed that produced by damaged knife edge. So, what is shutter? It is a resonance phenomenon that is caused by an interaction between at least two elements of the microtome knife block system, and force that cause shutter at maximize as the resonance. The shutter lines appear in the section at regular interval. To get an optimum section, three elements must be in correct position. Edge of the sample, block face are opposed to the knife edge and the clearance angle. Disturbance of any one of the three elements may cause sectioning artifact as incorrect sample rotation, incorrect knife angle, and incorrect sample angle. In this electron micrograph, we can see a narrow band of shutter run through the whole section from the beginning to the end, caused by localized defect in the knife. Another example of shutter, but this micrograph has a wide band of shutter. Also here, a shutter caused by a localized defect or a dull part of knife. While here in this electron micrograph, two parts in the same section of greatly different thickness are noticed. Moreover, two sections of greatly different thickness are lying side by side can be obtained by improper sectioning. During a collection of ultrasonic section on water baths, multiple section clusters or overlapping on the water surface may occur. Multiple sections cluster may lead to folding of the sections. Here we can notice that there is two sections overlapping each other, which is caused by aggregation of the multiple cluster of sections. While here we can see cracks that caused by folding of the section that was not enough to stretch or manipulation. Staining artifacts. Staining artifacts. Some researchers use old stored stain that decrease the resolution and contrast of image. So you should use freshly prepared stains. In this image, all the stored lead solution was used. Notice the resolution and contrast of the image that was greatly enhanced with freshly prepared one. Furthermore, some researchers may keep the sample for longer time in the stain that cause precipitation. So you should rinsing thoroughly but gently after the staining, which is mandatory to remove excess stain. Electron micrograph, we can see lead citrate precipitate and uranyl acetate precipitate. Irregularly shaped dark rings is produced by insufficient washing during contrasting procedure. Post-staining is an important step to enhance the contrast of the image, so skipping this step leads to hazy image with poor quality. It is a hazy image with poor quality, with no post-staining, which was greatly enhanced by post-staining step. Imaging artifact, inappropriate electron beam speed, beam damage or burn, or beam contamination. Beam damage or burn cause holes produced by other knife marks that are enlarged by heating of the electron beam. Hydrocarbon contamination was produced from focusing the beam too long on a single spot on the specimen, while contamination within the microscope may interact with the beam to produce rounded dark spot on the specimen, termed beam contamination. In this electron micrograph, we can see a heavy spot of contamination on the section was melted by the beam and vaporized on selected pattern over the section, illustrated by arrow. As regards scanning electron microscope, causes of artifacts, researcher, and processing and imaging like transmission electron microscope, but here we have another cause, which is sample. Some researchers, during obtaining the luminal sample, didn't wash it well, so proper washing by syringe or pipette preserve your sample and prevent sticking of the particle on the surface of the sample. Notice here food particle on the surface of the sample. Artifacts due to the nature of the sample. Charging artifacts. They occur when non-conductive or poorly conductive samples accumulate electrons within the sample, forming a charge. This results in bright or dark region, streaking or image distortion. They may be represented in many samples as ceramics, polymers, or biological samples. Horizontal bright and dark bands are a result of charging of a small hive beetle, while here in the other image we can see extensive charging artifact of human macrophage. So charging artifact is caused by non-conductive or poorly conductive sample. So the solution will be coating with thin conductive layer of gold, platinum, or carbon, or reducing the accelerating voltage using a low vacuum mode to dissipate the extra charge. Edge artifacts. They arise when a secondary electron emission is enhanced at sharp edges or rough surface due to increased electron escape probability. This creates artificially bright edges, rims, or shadow-like features that can be mistaken for the composition of the sample. It is common in fractured sample or those with pronounced surface roughness as fractured metal alloys or nanowires. If you work in fractured metal alloy, the edge effect may lead you to think that grain boundaries are enriched with precipitate. Here in this electron micrograph, we can see the bright edge is very clear. Edge artifacts, they are related to sample with pronounced surface roughness. So the solution will be rotate the sample or adjusting the detector position that may reduce the edge brightness. Sample degradation. Accelerated electrons interact within the sample and when analyzing pin-sensitive material, this interaction can damage and degrade the sample. These sensitive samples include non-conductive, organic, biological, and sample containing polymers. The degradation of sample can be seen in the form of cracks on the surface. Processing artifacts. Drying point artifact. It is a structural distortion that occurs in biological or soft specimen during drying. It's caused by surface tension when water or dehydrating solvent transition to gas, leading to shrinkage, flattening, or collapse of the cellular structure. With proper drying point technique, the sample preserves its original structure, but it becomes stretched and flattened by improper drying point technique. Sample contamination. It is a frequent artifact that appears as dark growing patches or layers on the specimen surface, gradually obscuring fine surface details during image. It is caused by hydrocarbon deposition that originate from gases or organic material in microscopic chamber that become fixed on the sample due to interaction with the focused beam. Also, poorly cleaned sample or bare handling of the sample may cause contamination. Here we can see bacteria and cellular debris over the surface of the specimen. Astigmatism is a common imaging artifact in which the electron beam doesn't converge properly, resulting in image distortion along one axis, which can change the orientation with focus adjustment. When an image is in focus and has no astigmatism, the high-energy electron beam forms a single point on the surface of the specimen. But when it's defocused, the beam is no longer form a single point but a round spot, so image become blurred. But if the spot is stretched in one direction, I mean that the spot is an elliptical spot, so the image has astigmatism. So what is the importance of identification of artifacts? Ensuring accurate interpretation, as artifacts can mimic or obscure the true structure of the tissue. Avoid misdiagnosis as artifacts can look like disease-related changes as shrinkage mimicking necrosis. Also, identification of artifacts preserves the scientific integrity of publications and report by reliable and reproducible data. Finally, it improves sample preparation and imaging techniques. Take-home message. Understanding how artifacts form and identifying their different types can change the interpretation of the morphology of the tissue and the reliability of your data. Taking steps to minimize them is necessary to make studies and research utilizing transmission electron microscope and the scanning electron microscope more scientifically sound and honest. For further reading, here are the references related to this topic. Thank you for watching. These are my accounts on ResearchGate and LinkedIn, and even you can email me on my official email for help. Waiting for your question.